Good afternoon, everybody. This is Liz Tran from Quasi. Welcome to the last Cyberseminar, part of the Cyberseminar series, Heterogeneity, Complexity, Anomalous Transport in Hydrologic Systems. Please welcome Diogo Bolster, University of Notre Dame. He is the host of the series.
Hi, everybody. Welcome to our last and hopefully most interesting of the seminar series, which will be given by Pietro de Anna, who has kindly agreed to stay up well past his bedtime. He's calling in from Switzerland, so I won't take too much more of his time. Pietro, whenever you're ready, the floor is yours.
Thank you very much, Diogo. I'm going to talk about biological and chemical activities in confined flows, and in particular about the role of heterogeneity and segregation. On the left, I am showing here an image, a schematic of a layer between the soil, the aquifer, and the bedrock. In this environment, many processes are taking place, like single and multi-phase flows, mixing between different chemical species, and bacteria. They can be present and they can swim around and be transported by the local flows, and they can grow, building biofilm. I'm going now to start with a simple example in order to introduce my point of view on how the small-scale processes, such those I am showing you, may impact a larger-scale behavior. Let's consider the chemical reaction that happens between two species, let's say A and B. In the left schematic, I'm showing the volume of the system where blue, green, and red particles they can interact, and basically if they meet, so when a reaction takes place, they can be transformed in some product. A simple model based on the mass action law would assume that the system is well mixed. So in other words, all the individuals, all the red and the blue particles, they have the same chance to meet each other. Under those hypotheses, the overall concentration of the chemical A, for instance, is following the equation on the right. So it's following the mass action law, which predicts that the rate of reaction is proportional to the product of the involved reactant concentrations. So for this type of system, if we consider that the initial concentration of the chemicals is the same, we can solve analytically this system of equations, and we can get a decrease for the reactant A that would follow the power law 1/time. But what happens if we consider a spatially extended system? In the image on the left, I am now discretizing the system in bins where the chemicals are inside. So now each of these bins can be assumed to be well mixed. So then, in order to consider the chemical reactions, we have to, the overall mass conservation, we have to consider the chemical reaction, but also the mass transport of the chemicals between one bin to the next one. So let's, for instance, consider that the mass transport is given by the molecular diffusion. In this case, this simple system of equation cannot be solved analytically. So if we run a simulation and we look at the result of this system, we will see that only for early time, the system would follow the kinetics predicted for a well-mixed system. For later time, what we observe is a deviation from the 1/time law towards T to the -1/4 law. So how can we understand this system? The point is that the overall reaction kinetics is limited by the mass transport. In other words, by the mixing. So in fact, if we look at the phenomenology of the system, if we look at what happens, for instance, at this early time, we see that the number of particles is pretty homogeneously distributed along our spatially extended system. But as long as the reaction takes place, the chemicals get segregated into islands of reactant, blue or red. So in practice, what happens is that the particles in an island of blue particles, they cannot react with the red particles. We have to wait diffusion to make the blue particle to diffuse towards an island of red particles. So in order So this was just the phenomenology of the system. Let's see how we can use this understanding in order to predict the behavior observed at a larger scale. The average concentration of one reactant, let's say the A, is given by the average concentration of the islands times the number of islands that we have in the system. So the average concentration of an island will decrease like time to the -1/2, just because by diffusion the island will stretch, and so they will occupy a larger volume, and so their concentration will decrease. But also the number of islands will change in time, because they will diffuse and they will meet each other, and so they will disappear when they meet a bigger island. So this means that the product of these two quantities, the concentration of the average concentration in the islands times the number of islands, give us this t to the -1/4 scale. But actually, this reasoning can be extended to any physical dimension, and we obtain the same result. So my point here is that for spatially extended system, it is very important to consider the mass transport mechanism in order to capture the overall kinetics, so the transformation rates. And since the subsurface is characterized by the motion of fluids within confined and very heterogeneous media, we have to consider this type of segregation in order to understand the overall transformation rates. So first of all, I would like to introduce my point of view on flow heterogeneity. And to do that, I will show you a very simple example of a turbulent flow, where it's clear the nature of the heterogeneity. In a turbulent flow, like a river or the ocean, the fluid flow is very far from solid boundaries. And therefore, the flow is dominated by inertia, so the velocities are typically high, and the fluid flow is characterized by eddies. If we take a cross-cut of this video, we would see something like that, a distribution, very heterogeneous, of the concentration of the tracer that at the beginning was only in the middle of our system. The typical interpretation of this type of system is that there is an heterogeneity at all scales. But what happens is that below a given scale, which is the Kolmogorov scale, basically we have just a simple shear, which will limit the heterogeneity of the system. And for oceanic type of flows, this scale, which is given by the energy of the flow and the viscosity of the liquid, is on the order of the millimeter. So the classical view is that from the millimeter above, we have heterogeneity. What happens about flow through confined spaces? These flows, they are typically close to boundaries, so they are dominated by viscosity, and therefore the flow is typically laminar. The classical interpretation of that is that the heterogeneity do not happen at small scale, but only at the scale where a geologic formation will vary, so at meter or above. So here I'm showing an old experiment performed at the Heidelberg University, where a tracer is injected from the top, and we can see that in this sandbox, this tracer is going down. The typical scale that we consider to be the heterogeneity of the system is the spreading length, which represents the length at which the system varies in space. But if we could zoom within the grains of this system, we would see something like that. So the concentration of the transported chemical is not well mixed within the pores. So what's about reactions? If we consider the reaction between two chemicals, one pushing the other in a relatively homogeneous system, so with homogeneous flow, we can assume that the total mass will evolve according to the mass transport at the interface between the two chemicals. And if the mass transport is given just by diffusion or dispersion-like mechanism, we can conclude that the overall mass produced by the reaction will evolve basically like the square root of time. But what's about mixing? The mixing is the ensemble of processes by which two or more given substances that are originally segregated, they tend to occupy the same volume. In the image on the left, we can see two ink drops that are originally segregated, and with a stick, they get stirred, and then they get mixed So a mixture can be considered to be a transient state between an initial segregation and the ultimate homogeneity of the constituents. Actually, this idea was quite well known to the humankind since at least 2,500 years ago, where mixing was considered to be the complete compenetration of objects. So nowadays we know that the mixing is ultimately done by molecular diffusion, so the thermal agitation of particles. So if we consider a system that is not homogeneous, like this one I'm showing now, we have to wait that mixing will be done by diffusion at the interface between the chemicals. And as we can see, the pores are not well mixed. So how can we predict the scaling of the mass of the product of the reaction? In order to answer this question, here we run some numerical simulation. I am showing here the concentration field of the reaction product between an invading chemical and a defending one. If we repeat the simulation for the same flow field, but increasing the molecular diffusion, you can see that the concentration field will be always heterogeneously distributed in the system, but its spatial extension will change. So in particular, if you look at the overall mass produced, so the dotted lines here represent what we would expect with an homogeneous model. So where the mass of the product would scale like the square root of time. In reality, what we see is that the mass of the product will scale in a quite weird way. So we can see that there are two regimes: an early time regime, where the mass produced scales very fast, and in a weird way with respect to the molecular diffusion coefficient. And at late time, surprisingly, all the different simulations, they collapse to the same behavior, which looks like independent on the diffusion coefficient. So the reaction kinetics in this case is given by the diffusive flux across the reactant interface. So this is equal to basically the integral along the interface of the diffusive mass flux. So the diffusion coefficient times the gradient of the concentration of the reactant. The idea here is to try to model the concentration of the reactant and the length of the interface between the solutes that seem to control the production of the mass. In particular, the concentration, the average concentration gradient is given by the ratio between the concentration difference between the left part and the right part of the interface, which is C0 minus 0, so basically C0, divided by the thickness of this interface, which is indicated by the letter S. So in order to model S, we have to consider that if we consider only advection, so the volume will be conserved. And this means that the product of the length sigma of the interface times the thickness of the interface, sigma S must be constant. So as long as the flow is going on, the length of the interface will increase because the flow is deformed by the presence of the grains. And therefore, the thickness of the interface must decrease due to this volume conservation. But if we consider also diffusion, of course, this interface must increase due to diffusion. So putting these two things together, we get a dynamical system, an equation for the evolution of the average interface thickness S, which we can solve and have this complicated shape. But if we put back this into our equation, we get exactly the good prediction for what we observed. But this is true only until the fingers of the invading reactant are not interacting one to each other. When this happens, we have to change model. We have to evaluate the length of the interface in a different way. In particular, we have to first observe that the fingers are now merging to each other in order to form larger bundles of, so larger aggregates of fingers. So we evaluate the length of the interface sigma as an average bundle length, L, times the number of these bundles, N. The length of each bundle can be approximated by basically the advective spreading, so the spreading of the fluid particles in the system. And the number of these bundles is supposed to decrease as long as these lamella, they start to coalesce. And so this is reasonably approximated by the available space, so 1 over S. And therefore, we obtain for the overall mass, basically an equation which is surrounded by the box, for which the mass is scaling like the advective spreading and do not depend on the diffusion coefficient. So the reactive interface is basically controlled by the flow heterogeneity, which in this case I represent as the spreading of these advected particles. So the question now is, okay, but what controls the pore scale flow heterogeneity? In order to answer this question, we consider a disordered arrangement of grains that we build with a Delaunay triangulation, where each triangle corresponds to the body of a pore, and each edge defines a pore throat. So let's prescribe, for instance, that the pore throats are statistically distributed as a power law, and we call the size of a pore throat lambda. We can generate then several distributions of grains which are distributed according to a power law. We then solve the Stokes flow within these geometries, and here on the left are shown the solutions for the different geometries in a linear scale, while on the right I'm showing the same solutions but plotted in a logarithmic scale, in order that the white color will represent the average velocity, so the red will be the velocities that are above the average, and the blue will represent the velocities below the average. As you can see, the flow field is characterized by channels of high velocities and zones of stagnations, where the flow is very low, up down to 4 orders of magnitude smaller than the average velocity. If we look at the distribution or the statistical distribution of these velocities, we see that the small velocities, so the ones that are below the average, they are distributed as a power law, which are characterized by this exponent alpha. And each simulation results to have a different characteristic exponent alpha. So how can we understand this distribution? Here, the idea is that we understand the flow in the pores like the flow through a pipe. So basically characterized by the proximity with the grain's walls. As represented here on the right, the flow through each pore is basically as a typical parabolic profile, which is the Eigenpoiseuille flow, which is given by this equation. And since we know the spatial distribution of the flow, we can reconstruct the probability distribution of the velocity to have a given value u within a single pore. Since we understand the PDF of a single pore, we can represent the overall flow distribution as a weighted average of all these pipe distributions. So doing the calculations, we find this result for which we can see that when the velocities are less than the average, basically the integral on the right is a number, and so the overall PDF of the velocity scales like minus beta half, where beta is the statistical distribution of the pore throats. And this happens to match exactly what we observe from the simulation. So in the inset on the left, I am showing the fitted exponent versus the theoretical exponent beta half. So far I discussed the role of the segregation on reactant in a diffusion limited system, about the reaction that takes place at porous media front, and on the origin of the flow heterogeneity that may control those displacing fronts. So what about microbe transport in a porous material? So bacteria are prokaryotic microorganisms that are ubiquitous in soil and in the underground. Typically, they are of few microns in size. They can be non-motile, so organized in colonies, also known as biofilms, or they can be motile, powered by flagella and or by their pili. They are very abundant, as I said. So in the air, we can find about 1,000 cells per cubic meter, while in open flows like in the sea, we can find about a million of cells per cubic centimeter, so per milliliter. But it's in the soil where we can find the most of the abundance of bacteria, which can reach up to 1 billion of cells per cubic centimeter. So bacteria have been observed several centuries ago by the introduction of a very important tool, the microscope. Without a microscope, we can see bacteria, as in the movie on the left, only as a population, as they appear when they grow a population of a few million of cells. But if we look at them with a microscope, we can see them individually and track their growth. Or we can also track their motion, as in this movie, where the red streaks represent the trajectory of bacteria tracked by a microscope. So now I'm going to talk about chemotaxis of microorganisms, which is the cell migration oriented along chemical gradients. So a population of bacteria, which are motile, they can swim randomly around, but if there is a chemical gradient in their environment, in some case they can swim randomly, but as a population they can drift towards the chemical gradient, like represented in the image on the right, where we do an experiment where we put a prescribed concentration of a given sugar on the right and no sugar on the left. And we see that in a time scale of the order of a minute, the bacteria, they randomly swim, but they drift towards the sugar. So the question is the chemotaxis relevant in flows through porous material? So can small-scale chemical gradients taking place between the grains in a porous material at the displacing front trigger this mechanism of chemotaxis? And if so, is chemotaxis relevant for macroscopic transport of bacteria? In order to answer these questions, I performed some experiment using microfluidics and video microscopy. This is an image given by a microscope where you can see a random distribution of grains, this disc and this moon-shaped object, and the little dots, the white dots, are bacteria. So we can zoom in and we can see and track the individual bacteria in this spatially extended heterogeneous porous material. So what we see is that if we inject a fluid to displace bacteria, we see that they get displaced in a non-homogeneous way. So there is some leftover of bacteria behind the average front due to the flow heterogeneity. But now, what happens if we inject a chemoattractant or a chemorepellent? So a solution that may attract or repel the bacteria population. So these are two movies that I'm going to show you now, where bacteria are swimming around while displaced by a fluid flow from the left to right. So in the left movie, we are injecting a repellent. As you can see, bacteria get displaced, but they also get accumulated in stagnation zones. And this accumulation can be quite persistent. If you look at the time scale, it takes about 300 seconds in order to clean this stagnation zone. If we inject the attractant, as you can see, the flow, of course, will accumulate bacteria in low flow zone, but these accumulations will explode, will stop very fast because of the swimming of the bacteria. Probably this will be more clear if I play the videos together. So now we inject the two together, as you can see, and the right movie, the accumulation of bacteria will explode in a much shorter time than the left one. So is this accumulation due to the swimming activity of bacteria that swim against the gradient of the injected chemicals relevant for their transport? So what we can measure from this type of experiment basically is the residence time of the bacteria in the system. We can see that while injecting a chemoattractant makes the residence time of bacteria homogeneous, so they spend on average the same time everywhere. If we inject the chemorepellent, they tend to stay in the stagnation zone for a much longer time than in the rest of the medium. And these stagnation zones, as you can see from here, correlates very well with the fluid residence time. So basically 1 over the local velocity of the flow. So what we observed is that there is an accumulation of bacteria. So due to the existence of flow-driven structures, and then the disruption of this accumulation due to chemotaxis, which imply the existence of small-scale, so sub-pore gradients. And also these impact the residence time of the microorganism within the host medium. But this observation also raises some questions. So, are the sub-pore gradients persistent enough in order to make the chemotaxis possible? We see that the chemotaxis is active for about 3 minutes at least. But if we consider the spatial scale of this pore, for instance, we would expect that molecular diffusion could destroy the gradient within a bit more than 10 seconds. So how can this be possible? And then, can we predict the total biomass present in the system? And can we understand the impact of the chemotaxis on the total biomass present in the system? So first of all, to understand the persistence of these gradients that trigger chemotaxis, we perform another experiment where we inject a tracer that is visible and that has the same diffusion coefficient as the sugar that we use as an extractant. As you can see, there are small-scale chemical gradients even for large times. And in particular, we can quantify this plotting the concentration gradient residence time, which is defined as the time in seconds that a given pixel is covered by a chemical gradient for more than that is higher than a given threshold. That is the detection threshold of a gradient. So basically, because of the mechanism that I discussed before, the gradients in the pore, they persist for a long time because the heterogeneity of the flow is stretching the front. And this stretching, as a result, leads to a persistence of the average gradient by winning against the diffusion. Here I am plotting the same movie as before, but on top of the residence time of the gradients. As you can see, these gradient residence time are acting as walls for the accumulation of bacteria. So in practice, the heterogeneity of the flow at small scale in a porous material can trigger the chemotaxis of the bacteria, and that can be responsible for their accumulation for a long time within it. So let's see how this will impact the microorganism removal in the porous medium. If we consider a simple system where the displacement is represented by an advective dispersion model, basically the projection of the bacteria concentration along the longitudinal direction, so the main flow direction, will be given by an error-like function as written here. So the total biomass in the system will be just the integral of this longitudinal projection, P. So the total mass of bacteria is given by this complicated expression below, which can be simplified into this exponential decrease 4 times larger enough. In fact, if we look at the total mass of bacteria that we can measure from our experiments, we can see that there is a decrease, an exponential decrease of the mass of bacteria. Please note that the vertical axis is logarithmic and the horizontal one is linear. So here different symbols represent different replica of the system. So if we average over all these replicas, we see that there is a clear exponential trend of this of the biomass that is in the system. And if we inject the trypton, which is the attractant, basically bacteria get removed much faster than injecting basically the same medium where they are, which act not as an attractant. So in practice, at the same time, we remove even 10 times more bacteria if we inject an attractant. So my research basically used theoretical and numerical modeling in order to understand the role of heterogeneity and segregation in in a confined structure like the underground, and laboratory experiments in order to observe and model processes like biological activity that we cannot model without experiments. So with this, I finish my contribution, and I'm very happy to answer questions.
Hi, everyone. If you have any questions, please type it into the questions chat box, and then we will read it out loud.
Pietro, can I ask, while other people are maybe thinking of questions, let me ask you one question. So your experiments, I mean, they're beautiful experiments at the end with the bacteria. Can you give some practical context in which what you're observing or what you're seeing might be important?
Well, for instance, if you want to design some bioremediation technique, you may want to know how the microbes that you are using they are going to invade your system, or how can you remove them. So here the idea is that the mechanism of swimming along the gradients of along gradients of chemicals that they can find in the soil or that you can inject can change by far their behavior in the system. So this may be important to take into account if you want to understand how long it will take to remove some bacteria from the soil, or how long it can take to inject them.
Thank you.
Any questions out there?
A quiet group. Last week there weren't any questions either.
I know. I guess I'll give a few more minutes, and while that happens, I will actually show my screen. See. Everyone can see that. We have another Cyberseminar series coming up. So after this one ends, we have another one that starts next week. It is on the hill slope hydrology and earth system models, and it's a collaboration between Quasi and NCAR, and so they are working on a synthesis paper, and they just want to do some presentations and start working on that paper. And so it's free to everyone to join, and you must register for it. And the first one starts on Friday, April 21st at 12 p.m. Eastern Time. So, all right, well, looks like we don't have any questions. But if someone does think of any questions, please feel free to email us. I will put our email address in the chat box. It's commanager@quasi.org, and then I will definitely forward it to Pietro if you have any questions after the Cyberseminar. All right. Diego, Pietro, anything you want to say?
I just want to, Pietro, thank you very much for that talk. I certainly enjoyed it, as I know hopefully everybody else did. Thank you for staying up as late as you have, and thank you everybody for attending the entire series. I hope you enjoyed it, and I look forward to seeing many of you at whatever conference we'll be to. Bye.
All right. Thank you, Diego, Pietro, and have a good day, everyone. Have a good weekend.
Thank you very much. Have a good weekend to you too. Bye-bye.
All right.
Bye. Thank you very much.